Greetings everyone! In this video we will explore microbiome analysis in R using microbiome package. Microbes are essential to our health, and studying them helps us understand diseases risks like colon cancer. We will use microbiome package to analyze 16 sRNA gene data and profile gut microbes, particularly focusing on their role in colon cancer risk, influenced by diet. A diet swap study showed how change in fiber intake altered the gut microbiomes in African Americans and Africans. Without further ado, let's get started. The microbiome package in R works with the phyloseq data format, which includes several key components. For example, OTU table, which has the abundance of microbial species in each sample. Sample metadata, information like age, BMI, and sex. Taxonomy table, phylogenetic trees, showing relationships between OTUs. In short, OTUs represent the distinct species present in each sample. We will be using example data from the microbiome package already stored in phyloseq format, so no pre-processing is needed. Firstly, we upload our libraries. We have library microbiome, the player, phyloseq, GGPUBR, complex heat map, or color brewer. Let's run our code. Then we upload our data. So data, diet swap, and then diet swap to look at our data. Let's run our code. As you can see, we have OTU table, which contains 130 taxa and 222 samples. This table holds the counts of operational taxonomic units or OTUs for each sample. Then, sample data, 222 samples with 8 sample variables. This contains metadata associated with each sample, like for example groups such as lean, overweight, and obese, time point, and nationality. And then, taxonomy table, which contains 130 taxa classified across 3 taxonomic ranks. This table describes the taxonomic classification of each OTU. So regarding OTU table, it contains 130 species, taxa from 222 samples. The columns here indicate samples from the experiment, while the rows indicate operational taxonomic unit or OTUs. Each sample here is a community of microbes that was sampled from a person's mucosa that undergoes the diet swap treatment in a specific time point. The number in each column indicates the total number for each species for each sampling. There are 130 species that may be present in the sample. Now let's inspect the first 10 microbes, OTUs, for the first 7 samples in the dataset. So, we write diet swap at OTU table at dot data from 1 to 10 and 1 to 7, and then filter data dot frame. Let's run our code. As you can see, the output displays the counts of operational taxonomic units for the first 10 microbes across the first 7 samples in the dataset. The first column here lists the names of various OTUs such as Actinomyces cti, Aerococcus, Aeromonas, and so on, while the subsequent columns represent the abundance of these OTUs in each of the 7 samples. So we have sample 1, 2, 3, 4, 5, 6, and then 7. And for example, the OTU here, Akkermansia, shows counts of 18, 97, 67, 256, 21, 16, and then 26. This indicates that this microbe is relatively abundant in certain samples, particularly in sample 4, where its count reaches 256. On the other hand, Aerococcus here has a count of 0 for all samples, indicating its absence from the dataset, suggesting that this specific microbe is not present in the sampled individuals. Now let's inspect the metadata associated with our dataset. The metadata provides detailed information about the sample data, gives us insights into how the experiment was conducted. It offers a glimpse into the research design, including the number of participants and the time points used to collect the complete dataset. So we write metadata and then meta, and we put our data, diet swap, and then head to inspect our data. So head metadata. Let's run our code. And here we have it. So as you can see, here we have samples, sample 1, 2, 3, 4, 5, 6, and then subject. The subject unique identifier for each participant. So sample 1 BIN, sample 2 NMS, and so on. After that, sex. We have male and female. Then nationality. We have AAM and AFR. The AAM for African American and AFR for Native African. It's important for dietary and microbiome comparisons. And then group. General classification of the subjects indicating their dietary or treatment group. Then sample, then time point, and time point within group. The time point, integer values representing when samples were collected. It's vital for tracking changes over time. And time point within group, sequence of time points within each group. It's useful for temporal analysis. Then we have the BMI group. It categorizes subjects based on their BMI. So we have lean, overweight, and obese. As you can see, obese, lean, overweight, obese, and so on. It's crucial for examining health impacts. Now we should correct the data types in the metadata, because when we analyze data in R, it's crucial that we tell R what type of data we are dealing with. In our metadata, we have two columns, time point and the time point within group, which represent the categories. These categories indicate different time periods when samples were taken. But why it's important? Firstly, these two variables, time point and time point within group, are not numerical values. They represent categories or groups like group A and group B, or different time points like week 1, week 2, and so on. Telling R that these are factors helps it understand that they represent groups rather than continuous numbers. And secondly, to correct the analysis. If we leave them as plain numbers, R might treat them as continuous data, which would be incorrect. This could lead to wrong conclusions when we analyze our data, because R might try to perform mathematical operations on them, like finding averages, for example, which doesn't make sense for categories. And lastly, for visualization. When we plot the data or create graphs, specifying these variables as factors allows us to categorize the data correctly. So, let's correct our data types. So we write metadata, and then metadata, filter, mutate, time point equal as factor, time point, and then time point within group equal to as dot factor, time point within group. Let's run our code. And then let's look at our data by using summary. As you can see, we have subject, lists unique subjects and their counts. For example, we have AZH, it appears 6 times, indicating multiple samples for that individual. And then sex, we have female and male. It displays gender distribution within 102 female and 120 male. And then nationality, AAM 123, AFR 99. As we said before, AAM is African American and AFR is African. Then group, it shows the number of samples in each group. For example, here we have 72, and then 75, and then 75. It's important for understanding experimental classifications. And then we have sample. Lists sample identifiers within a total of 222 samples. Then time point indicates the number of samples collected at different time points. For example, here 38 at time point 1. It's useful for temporal analysis. And then time point within group provides a detailed breakdown of samples within each group. It's essential for tracking changes over time. And lastly, BMI group classifies individuals based on BMI. For example, 56 lean, overweight is 76, obese is 90. I'd like here to mention that the diet swab treatment were divided into 3 stages groups. There are 6 time points in the experiment. In case of ED, initial endoscopy as a microbial community profile, while on their usual diet before diet swab treatment. HG here, its home environment, while on their usual diet prior to diet swab. And then DI is a dietary environment while on diet swab treatment. Now we have inspected and understood the OTU table and the metadata. We will retrieve the taxonomy table using this command, which is taxon equal to tax table diet swab. And then we look at the taxon by using head. Let's run our code. The taxonomy table allows us to see how each operational taxonomic unit, or OTU, is classified into higher taxonomic ranks, like phylum, family, and genus. Great! So now what's taxon? Taxon is the name of the microbial species or group. It indicates the name of the microbial species or group. But how this output is important? The presence of multiple phyla, like actinobacteria or proteobacteria, it indicates a diverse microbial community, which can be important for various ecological functions and health implications. We can combine the 3 components into a data frame using PSMelt function. Applying this function to the phyloseq object has several advantages. First, it transforms complex multi-dimensional data into a simpler 2-dimensional format, making it easier to see the relationships between samples and their OTUs. Secondly, TD format allows for straightforward filtering and manipulation with R tools, like the player, enabling quick examination of specific groups or conditions. And thirdly, TD data is more compatible with visualization tools like ggplot2, resulting in clearer plots for displaying microbial abundance across samples. Finally, melted data retains essential sample information, such as sex, nationality, and health status. So, let's use our function psmelt. So we write diet, psmelt, diet swab, and we look at our data with head. Let's run our code. And here we have it. As you see, the output data frame consists of OTU, sample, abundance, subject, sex, nationality, group, sample, time point within group, BMI, phylum, family, and genus. OTU, names for operational taxonomic units. For example, Provitola melaninogenica, indicating the microbial species present. And then sample. Here, identifiers for each sample. For example, sample 208, here showing where the OTU was detected. And then abundance, the count of the specific OTU in each sample. For example, 14,270 in sample 208. And then subject, unique identifier for the subjects from whom samples were collected. And then sex, the sex of each subject, male and female. Then nationality, like African American or African. And then group, classification of subjects into different groups based on the criteria, like dietary intervention, ED or HE, and so on. Now let's dive into some exploratory data analysis using the plotting functions from the microbiome package. Now let's create a visual representation of the proportions of different BMI groups across nationalities using this code. This step is important for exploratory data analysis or EDA, as it allows us to quickly assess the distribution of BMI classification, such as lean, overweight, and obese among various nationalities in our data. So we write plot frequencies, x equals sample data, we put our data, diet swap, and then groups, BMI group, factor, nationality, to make this analysis based on the nationality, which is African American or African, and then labs fill equal nationality to make different colors, theme minimal, and theme axis text equal element text. Let's run our code. And here we have it. We have here the y-axis proportion with percentage, and the x-axis here, it only indicates the BMI group, like lean or overweight or obese. So based on the plot, it's evident that African Americans, which are the red color or pink color, predominantly represent the overweight and obese categories. Here, overweight and obese, because as you can see, the red color here is more than the blue color. And also for obese. But for lean, it's oppositely. African is more than African American, regarding the proportion. In microbiome analysis, the primary goal is to explore and understand the composition of microbial communities within a given dataset. This analysis typically involves visualizing the microbial community profile, which helps identify the types and abundance of microorganisms present in the samples. A common approach for visualization is to create bar plots that display composition of microbiomes in each sample. But before proceeding with visualization, it's crucial to filter out only the prevalent taxa from the dataset. Prevalent taxa are identified as those that appear frequently across multiple samples. This filtering step ensures that only the most relevant and consistently observed microbes are induced in the analysis. So, let's filter our prevalent taxa. So, we write core diet, and then core x equal diet swap, detection equal 50, as a threshold for presence of each OTU in a sample, and prevalence 50 over 100. This is threshold for the fraction of OTUs that exceeds detection threshold. And then we look at our data or core diet. The core function is used to filter the dataset to retain only the prevalent taxa based on specified thresholds. We put our parameters, x equal diet swap. This specifies the phyloseq object containing the microbial data to be filtered. And then detection equal 50. This threshold indicates that a taxon must be present in at least 50 counts in a sample to be considered present. This helps remove taxa that are rarely detected and may be noise in the data. And finally, prevalence equal 50 over 100. This threshold means that a taxon must exceed the detection threshold in at least 50% of the samples to be considered prevalent. Let's run our code. So, as you can see, we have OTU table, 17 taxa, and 222 samples. This indicates that our filtered dataset now contains 17 operational taxonomic units or OTUs. These are the taxa that meet our specified criteria for detection and prevalence across 222 samples. And regarding 222 samples by its samples variables, the dataset still includes 222 samples, which means that the sample metadata such as subject IDs, sex, nationality, etc. is preserved for each sample. We will still be able to link any alphadiversity metrics or other analysis back to the associated metadata. And then 17 taxa by 3 taxonomic ranks. The taxonomy table is now limited to the 17 taxa that we filtered. This table categorizes each OTU into the taxonomic ranks, which may include phylum, family, and genus. This allows us to analyze and visualize our results at various taxonomic levels, enhancing our understanding of the microbial community composition. But why this step is important? By focusing on prevalent taxa, we are likely to improve the quality and reliability of our diversity analysis. Rare taxa may introduce noise and variability that don't reflect true biological patterns. Before diving into the analysis of microbial community, it's essential to prepare our data to ensure accurate and meaningful results. One common pre-processing step in microbial analysis is log transformation, which helps stabilize variance and makes it easier to compare different samples. We will apply log transformation to our filtered dataset using the transform sample counts function from the phyloseq package. This transformation will enhance the interpretability of our results by reducing the impact of highly abundant taxa and revealing the contributions of low abundance species. We write diet log, transform sample counts, and we put the core data, and then function x, and then log 1 plus x. Let's run our code. Now we can proceed to calculate alphadiversity metrics using this refined dataset. This will give us insights into the diversity within each sample based on the prevalent taxa that are most likely to be significant for our studies. In microbiome research, one key aspect we explore is alphadiversity, which helps us understand how diverse the microbial communities are within different samples. Think of alphadiversity as a way to measure the variety of microbes. Alphadiversity, it measures how many different species are present, which is the richness, and how evenly individuals are distributed among those species, which is evenness. For example, imagine you have a single garden with various types of flowers. The flowers are 10 roses, 5 tulips, 5 daisies, and 1 sunflower. So regarding the richness, there are 4 different species, which are roses, tulips, daisies, and sunflowers. Regarding evenness, roses dominate the garden, while tulips, daisies, and sunflowers are less common. This results in low evenness. So the conclusion, alphadiversity of this garden can be described as having moderate richness for a species, but low evenness when a species dominate. So key metrics of alphadiversity, observed richness, for example, and also we have Shannon diversity. For observed richness, if your richness score is high, it means that your gut microbiome is balanced with different kinds of microorganisms living in it. A diverse and balanced ecosystem is generally linked to a good health. as it supports digestion, nutrient absorption, immune function, and protection against harmful pathogens. On the other hand, lower richness score is a sign of a less diverse gut microbiome with fewer types of microorganisms. And regarding Shannon diversity, it doesn't only count as a unique species, but also considers how evenly the individuals are distributed among those species. For example, if one plant type dominates the garden while others are spores, the Shannon index would be lower than if all plant types were equally abundant. Higher Shannon index suggests a balanced community, where no single species is overwhelming. Now, let's go through our steps. We will calculate and visualize these alpha diversity metrics. So, reach, estimate richness, we put coordinate, measures, we want to look at observed, and Shannon. Let's run our code. As you can see, here is the samples, and here observed, and here Shannon. Regarding the observed, it's 17 in all samples. This represents the total number of unique OTUs, operational taxonomic units, present in each sample. So the value of 17 meaning that each one has the same number of unique bacteria. And for here, Shannon, this index considers both richness and evenness of the community, providing a more nuanced understanding of diversity. A higher Shannon index indicates greater diversity. So as you can see, the Shannon diversity values range from about 1.40 to 2.30. For example, sample 9 considered to have the highest Shannon value, which means it has more variety in its bacteria. On the flip side, sample 6 here, it has the lowest value at 1.40, indicating that it might be dominated by just a few types of bacteria. So even though all samples have the same number of unique bacteria, the different Shannon values tells us there are differences in diversity. Higher Shannon values mean there is a better mix of bacteria, which is great for gut health. Lower values suggest that only a few bacteria are doing most of the work. Now let's visualize our alpha diversity using box plots. As you can see, plot richness, coordinate, X equal BMI group, measures, observed, and Shannon, GM box plot, theme classic, and then the theme. Let's run our code. As you can see, because all samples have 17 types of bacteria, so they are all the same regarding the group lean, overweight, and obese. But for Shannon, the range of values are different, showing that some samples have a wider variety of bacteria than others. As we can see here, the obese group has the highest microbial diversity, which could mean that they have a richer and healthier mix of bacteria. And here, for lean group, has a moderate variety, while the overweight group here shows the least diversity, pointing to either fewer types of bacteria or less balanced mix. This suggests that the types of bacteria we have in our gut can play a big role in our overall health, including our risk for diseases like colon cancer. Regarding the lean group, the median Shannon diversity index is around 1.75, suggests that microbial diversity in lean individuals is moderate low. The box size, indicating the interquartile range or IQR, shows some variability in diversity among individuals in this group, but it's not extreme. And for the second group, overweight, it has a higher median, around 2, compared to lean group, suggesting that microbial diversity is slightly higher in overweight individuals. However, the small size of box suggests that diversity is more consistent among individuals in this group, with less variation between them. And finally, the obese group. The median is about the same as the lean group, 1.75, meaning the overall diversity is similar. However, the slightly larger box suggests that there is more variability in microbial diversity among obese individuals compared to lean group. So, plot richness, core diet, X BMI group. We can measure only the Shannon and we make color equal X. And then gene box plot, fill with sex, theme classic, basic crab sex, and then theme. Let's run our code. As you can see, we have female and male. Female are red, males are blue for lean, overweight, and obese and overweight, and no lean for female. So after preparing our microbial richness data, the next step is to assess whether there are significant differences in microbial diversity across the BMI groups. To do this, we can utilize the Wilcoxon rank sum test. It's a non-parametric statistical method suitable for comparing two or more groups when the data may not be normally distributed. We will first convert our richness data into a data frame for easier manipulation. Then, add the corresponding BMI group information. Finally, we will conduct the Wilcoxon rank sum tests for both richness metrics using false discovery rate method. So, rich data frame as to data frame, rich diff, we choose the BMI group, and then sample data, core diet, and then BMI group. Then Wilcoxon rank sum test for observed richness. Wilcox observed, pairwise Wilcox test, rich observed, each data frame BMI group, and p-adjust method is FDR, or false discovery rate. And then Wilcoxon rank sum test for Shannon diversity. Wilcox Shannon equal to pairwise Wilcox test, rich diff Shannon, rich diff BMI group, p-adjust method or FDR. And then we print our results for Wilcox observed and Wilcox Shannon. And then we print our results. Let's run our code. As you can see, we got our result. Here there is no data because there is no differences in our samples, as OR contains 17. And then p-value adjust method FDR, pairwise comparisons using Wilcox rank sum test with continuity correction. And then data rich diff Shannon and rich diff BMI group. We have overweight, obese, and the lean. Overweight for lean versus overweight is 0.0487. This indicates a statistically significant differences between the lean and overweight groups at the conventional alpha level of 0.05. This means that the diversity in the microbial communities between these two groups is likely different. And for overweight versus obese, p-value equal to 0.0021. Here, this is also statistically significant, indicating that there is a significant differences in microbial diversity between these two groups. And finally, lean over obese, which equal to 0.4385, suggesting there is no statistically significant differences in channel diversity between the lean and obese group. Now, let's go for betadiversity. This concept helps us understand how different groups of microbes are from each other. Betadiversity measures how unique one neighborhood is compared to other. In our study, we want to see how diverse the microbial communities are and how they differ from one another. In simpler terms, in betadiversity case, imagine we have 3 separate gardens, garden A, B, and C. Garden A have 10 roses, 5 tulips, 5 daisies, 1 sunflower. Garden B have 4 sunflower, 4 daisies, and 2 marigolds. Garden C have 1 rose, 1 tulip, 1 daisy, 1 sunflower, and 1 marigold. So regarding the comparison, Garden A versus Garden B, both gardens have daisies in common, but Garden A has roses and tulips, while Garden B has sunflowers and marigolds. They share one species, so they are somewhat similar, but they also have different species, indicating moderate betadiversity. But for example, garden B and garden C, these gardens have different species with no overlap, indicating high betadiversity as they are quite dissimilar. So to find patterns in our data, we will use a method called Principal Coordinates Analysis, or PCoA. This is a technique that helps visualize the relationships between different samples based on their microbial composition. Think of it as creating a map that highlights how closely related or distinct our microbial groups are. So, set.seed equal 100. It ensures that the random elements in the analysis can be repeated with the same results. And for ordinate function, here, phyloseq equal diet_log, method MDS, distinct array. The ordinate function creates an ordination object which using the diet_log data, and the Bray-Kurtz method is used to measure the dissimilarity between samples. And then we prepare eigenvalues to adjust the axis. We write evals equal to ord values eigenvalues. Evals equal to ord and then $sign values $sign eigenvalues. It retrieves the eigenvalues from the ordination to adjust to the plot axis. And then plotting. We use plot ordination, physeq equal diet_log, our data, ordination ord that we have created up here, and then join point size equal to, and then coord fixed square evals 2 evals 1. This ensures that the axes are scaled appropriately based on the variance explained by the data. Let's run our code. And here we have it. As you can see, we have axis 1 and axis 2. It seems to be no extreme outliers in our data. We can proceed with further analysis using this data. We can color our dots for better visualization. So, we add here color based on the nationality. Let's run our code. As you can see, it's now clearer. We have nationality African American in the red color and African in the blue color. So from the plot we can observe that the microbial composition is notably distinct between the African American and the native African, as indicated by the variable of nationality. This suggests that diet may influence microbial communities. Our study aims to investigate whether a diet swap can alter the initial microbial composition between these two groups. To visualize the differences in microbial composition among subjects of different nationalities, we will create a plot showing microbial abundance. To effectively understand and compare microbial communities, we often visualize them using stacked bar plot. Each bar represents a specific sample, showcasing the diverse array of microbes present and their relative abundances. This visualization allows us to identify which microbes dominate in each sample or across different groups. But before we create these plots, we need to change our original microbial abundance data into relative abundances. This means we will express the amount of each microbe as a percentage of the total number of microbes in each sample. So, diet relative, and then microbiome, transform core diet compositional. This line takes the original dataset called the core diet and transform it. But how does it work? By using the transform function from the microbiome package, we can change the data so that each microbiome's abundance is expressed as a fraction of the total microbial count in each sample. This means instead of showing how many of each microbe there are, It shows what portion of the total they represent. And then we make an inspect. Dot relative at OTU table at data 1.3 and 1.3. This line checks the transformed data by looking at the first 3 rows and the first 3 columns of the OTU table in dietary. So let's run our code. And here we have it. The data represents the relative abundances of different microbial species across 3 samples. We have in sample 1, sample 2, and sample 3. Each row lists a specific microbiome, for example, alphadiversity, and the Bacteroides fragilis, and the Bacteroides vulgaris. The number under each sample column indicates the proportion of each microbe within the total microbial community. For example, Bacteroides vulgaris here constitutes about 47.14% of the total microbial community in sample 1, indicating its dominance in that sample. In contrast, Bacteroides fragilis has much lower presence in other samples, as you can see. Now let's filter out our microbial abundance data to focus on specific groups based on nationality and BMI classification. By using subset samples function, we will create distinct subsets for each BMI category within both groups, African American and African population. So, for the African lean, subset samples, diet relative, nationality, African, BMI group lean. We make the same for overweight and for obese, and the same for African American, for lean, overweight, and obese group. Let's run our code. And now, let's plot. We use plot composition, African lean, for example, taxonomic level genus, average by time point, OTU sort, abundance, X label equal time point, and then labs X time point, and Y abundance, and we'll put a title, Native African lean. Let's run our code. And here we have it. We got our plot with many taxa. As you can see, each taxa represents a color. For example, here the red is the Prevotella melanogenica, and Clostridium for this green, and so on. So we have the abundance, and we have the time point. And now, let's plot the average microbial composition samples. So we will make a plot for African lean, and African overweight, then African obese, then African American lean, African American overweight, and African American obese. And to combine these all plots, we will use GGP UBR. We make GGArrange, plot African lean, plot African over, obese, and we make another one for African American, and we put the widths, and then GGArrange, we put here AF for the 3 plots of African, and AM, the 3 plots of African American, in row 2 labels, we put Native African and African American. So let's run our code. And here we have it. We got 6 plots. 3 for African, and 3 for African Americans, containing lean, overweight, and obese, lean, overweight, and obese.
And here we have reached the end of our analysis. I will leave the links to the resources in the description of the video that helps us to do this analysis. Thanks for watching, see you next time.